Hi guys, it's the science teacher here back with another video. This time I'm going to try and go through my predictions for the biology paper 2 exam. As I've said in my previous videos, I will not probably be 100% right on all my predictions. I got, I think for my paper 1, around 60-70% of the paper. So what I would say is with this video, also watch my end of topic videos in order to make sure you cover all bases. So let's have a look first at where I got my information from. I used the AQA's website and they've given out some advanced information on what they think is going to come up in the paper. And if you look for the foundation and higher, they are very similar. So this video will be covering both foundation and higher. And I'm going to definitely have a bit of focus on the required practicals and the bullet points where it says this is the major focus of content in the exam. And I have kind of omitted everything in the section which it says topics not assessed in this paper. The first required practical we are going to take a look at is how plants respond to a stimulus. Now, the main thing that they probably are going to ask you about is how a plant responds to light. And we call a plant's response to a stimulus tropism. And what I think they will use is, I think they'll use light as the stimulus, and we're going to look at how the plant responds to this light. Now, we say that plant shoots are positively phototropic, and that means that they grow towards a light source. And we can prove this fact by setting up the practical in this way. Here I've got different germinating seedlings, and I have done different things to their top of the root. Here I've put a cover on the top of my plant, my newly germinated seedling, and as you can see, what has happened to it is it has just grown vertically upwards. This plant cannot respond to the light source due to the fact it's got a black cover on, and therefore it just grows straight up. Here I've removed the top of my plant. Here I've done nothing to it, it's just exactly the same. And here I have a transparent cover so that the light can pass through it, just to prove that it's not the weight of the cover doing anything to the plant, it must be the light that is causing this change in the growth pattern. So as you can see, what happens is if I have removed the top of my plant, it no longer grows, it has stunted growth, and this must be to do with the plant hormones causing the growth. And if I remove the top, then it cannot grow any further. If I do nothing or have a transparent cover, the plant bends towards the light. And we're going to have a look at the next slide about what causes this bending growth. But just before we do that, I just want to talk a bit about a different type of tropism, and that is gravitropism. And here you can see that I've tipped a plant on its side, and you can see that the roots they grow in the direction of gravity. Even though the plant pot's on its side, the roots continue to grow downwards, and this is called geotropism, and the roots show positive geotropism. And if you look at the stem of the plant, you can see that it's growing away from gravity, and this is called negative geotropism. Now, just like humans, plants have hormones, and this is what causes those tropisms that we saw on the last slide. And the plant hormone responsible for growth are called auxins. Now, if we put the light source here, you can see what happens to my auxins. They gather up on my plant shoot away from the light source. And what this causes to happen in the plant is it causes excessive growth on this side of the plant, so it grows more. And because of this growth, it causes my plant to bend. And this is why we observed the pattern on the last slide of positive phototropism in the shoots, is because these auxins are gathering on the side furthest away from the light source, causing that side of the plant to grow faster, and therefore the plant bends towards the light. Now, roots actually do the opposite as well. If you were to shine light at plant roots, it would grow away from the root, away from the stimulus, and that's obviously called negative phototropism. And all that happens is the auxins gather up furthest away from that light source. There are other hormones that plants produce for different responses, so we're just going to look at them. We're not going to look at how farmers use them, as that's something that it's said specifically will not be on the paper, so I'm going to go through this quite quickly. So some hormones that plants can produce in order to get a certain response. One of them, of course, is auxins, and this is the growth hormone. There is also the hormones called gibberellins, and what gibberellins do is promote germination in seeds. And there is also a hormone called ethene, and what ethene does is it ripens the fruit. I don't think it will ask you about any other hormones, and this might not come up. I just wanted to put it in just to be safe. The next required practical to look at is how to investigate the populations of species. And the way to measure the abundance of different species is by using what we call quadrats. And quadrats can be used to measure the abundance of species by a few different ways. You can either count up how much of each species you've got. Maybe this is how you do it for a slow-moving insect, or what you can do is you can use the percentage cover of the quadrat in order to kind of make an estimate of how much of that species is present inside the quadrat. Now, what is important for when you are using quadrating to determine the health of a population is you need to make sure you avoid bias when placing that quadrat down. So we often use random sampling in a field in order to decide where we place our quadrat. And to do a random sample, you can use random number generators, or you can use dice in order to decide where you place your quadrats down. Now, this is the technique that you would use if you have the same type of habitat. For example, if you've got a forest or you have a large field, you could use random sampling to decide where you want to place the quadrat, and then you could finally calculate how many of each species you have in that field or forest. We're now going to get on to more of the content of the exam and away from required practicals. We are going to look at the central nervous system and how humans respond to different stimuli. So if we have a stimuli, which just means a change in the environment, well, what happens is we have receptors, and these receptors will detect the change in the environment and send it to our central nervous system. Our central nervous system basically involves our spinal cord and our brain, and this is what coordinates our response, and it sends it back to our effectors, and this is how we make a response. But how do these signals travel round the body? Well, they travel through electrical impulses through what is called neurons. So let's have a look at some of these neurons and what happens to the electrical signals. So over here, what I've just drawn is just a simple neuron, and we're going to look at some of the key features which makes it perfectly adapted for carrying these electrical impulses across our body. And firstly, let's have a look at these. They look like fingers attached to our cells, and they're called dendrites. And the reason why the neurons have these dendrites is so that they can make other neuron connections and therefore pass on the message, the electrical impulse that comes across. As well as dendrites, they have an incredibly long axon. And the reason why they have this long axon is so that there is less energy wasted from having too many connections. It can travel along one neuron, wasting less energy. When two neurons meet, there is a gap between these two neurons, and we call that a synapse. And what happens at this synapse is there's a small gap between the two neurons, and the electrical signal arrives at the end of one of these neurons, and it's converted into a chemical signal that can travel between the gap. And when this chemical signal arrives at the next neuron, it's converted back into an electrical signal to transmit quickly across the next neuron. Now, we've only looked at voluntary responses at the minute, but what happens if you don't have control over your response? What happens if that reaction needs to be so quick? We call these types of reactions reflex reactions, and they're around for safety purposes. Now, what happens in a reflex reaction is it misses out the coordination center, so your brain. The reason for this is because these responses have to be super quick. Imagine, for example, you touch something extremely hot. You want to move your hand away as quickly as possible. So how did these responses happen? Well, first, the stimuli needs to be detected, and we use sensory neurons to detect this stimuli. And for a reflex reaction, this stimuli is going to be quite extreme. Maybe you've been pricked by a needle, or maybe that you've touched something extremely hot and you're going to need to move away very quickly. After the sensory neurons, it travels down relay neurons, and we know now that the gap between these two different neurons is called a synapse. And this is kind of the key bit that's different to voluntary responses, is it no longer travels to the coordination center, which is your brain. It will just go straight back to your effectors, and it travels to your effectors via motor neurons. And the motor neurons are attached to your effectors, where you are going to have a response. Your effectors are usually going to be your muscles for a reflex reaction, so you can move away very quickly. Now, at this relay neuron, this is kind of the key bit in a reflex reaction. A response is also sent to the brain, however, it goes through the motor neuron quicker, and that's why you get that response without even thinking. And it's also important, if you do get like a 6 marker about reflexes, to talk about what's happening in the muscle to move away, and the muscle's often contracting in order for you to be able to move and get away from whatever hazard there is. Not all responses in our body are controlled by electrical impulses. Some of them are a lot slower and longer acting. And these are controlled by hormones. And what a hormone is a chemical messenger that travels around our body. Now, hormones are secreted from different glands inside our body. We have some glands in our brain, and the main one in our brain is called the pituitary gland, which is also known as the master gland. The reason why we call the pituitary gland the master gland is because it also produces other hormones, that can stimulate other glands to start secreting hormones. For example, the pituitary gland can produce a thyroxine stimulating hormone, and then the thyroid, which is situated here, will start producing thyroxine. And this hormone called thyroxine controls our metabolic rate and growth. In the pituitary glands of females, they also produce a lot of FSH and LH, which are important in the menstrual cycle. I know the menstrual cycle is one of them things which it says is not going to be assessed in this paper, so I'm not going to go into them in more detail. Then these two glands here, there's two of them, so I'll just draw two branches to show where I'm meaning. These are our adrenal glands, which produce obviously adrenaline, and adrenaline is what produces the fight or flight response. You might have felt before going on a roller coaster, really quite jumpy. That's because your body is producing a lot of adrenaline, and sometimes this can last a while. You might have noticed if you've come off that roller coaster that your heart is still beating quite fast. That's because of the fact that adrenaline is longer acting. It's a chemical messenger, isn't it? It's not just them electrical impulses. Then, depending on if you're biological male or biological female, you produce different hormones in your sexual organs. And here you can see on the biological male, there's testes, which produce the hormone testosterone. And in biological females, produced in the ovaries is estrogen. We're now going to look specifically at the pancreas and a couple of hormones that are released in our pancreas in order to control our blood sugar levels. Now, what happens is if our blood glucose level gets too high, our pancreas releases a hormone called insulin. And this insulin tells our bodies to do a couple of things. What happens is our cells start taking in some of that glucose that's in the bloodstream. And also some of the glucose is stored as glycogen in our liver. Now, this is really important and something that's quite hard to understand. Why do we turn our glucose into glycogen? Well, what happens is glycogen is insoluble, whereas glucose is soluble. So we can store it so it doesn't have to leave our body. And the way we store it is in glycogen, which is stored in our livers. Now, our blood glucose levels are never exactly zero. It's always going through this loop. So what happens if our blood glucose levels are a bit low? Well, our pancreas releases a different hormone this time. It releases something called glucagon. And you just have to remember that the hormone's glucagon, the store of glucose, is glycogen. And what this hormone does is it tells our bodies to release some glucose that's been stored. And also it tells our livers to convert the store of glycogen back into that soluble glucose. Now, unfortunately, not everyone is able to control their blood glucose levels as they would like, and we call this condition diabetes. And we're going to have a little bit of a look into what diabetes actually is and the two different types of diabetes. Now, diabetes can be split into two types, and we're going to look at the first one as type 1 diabetes. And what happens in type 1 diabetes is your body doesn't produce enough insulin. Now, because your body doesn't produce enough insulin, how we treat it is we usually give type 1 diabetes sufferers insulin injections. Now, you might be saying, how do people contract type 1 diabetes? Well, type 1 diabetes is often something that people are born with, and it is thought to be something that can be passed down through genetics. But what about type 2 diabetes, the other type of diabetes? Well, type 2 diabetes is when your body stops responding to the insulin. And what doctors do to patients that suffer from type 2 diabetes is they often try and monitor their diet and try and help with exercise as well. And with type 2 diabetes, it's something that can be developed during people's life. It's something often contracted through old age, and there is some factors that can increase your chance of contracting type 2 diabetes. And we call these risk factors, and obesity is a main cause of type 2 diabetes. A key question they could ask is asking you to compare and contrast type 1 and type 2 diabetes. So if they did ask you a question like this, what you'd want to be doing is giving the similarities and the differences between the two. We're now going to look at genetic characteristics, and we call things that control our characteristics genes. And a gene is just a short section of a chromosome which codes for a certain protein. And it's important to remember that in every adult cell, we have these chromosomes, and we have 23 pairs of chromosomes. We get 23 from our mother and 23 from our father. And what we can have to code for different characteristics are alleles. And alleles are different forms of the same gene. For example, brown hair and black hair would be alleles. So let's look at this woman over here and look at some of her characteristics and look at some of the alleles that could code for the proteins that show her characteristics. So this woman has black hair. Now, black hair is a dominant gene. And what I mean by that is because of the fact we get 23 pairs of chromosomes, one from our mother and one from our father, you only need one black hair gene in order to show that characteristic. So that means there's two sets of alleles that could code for the protein for black hair. So let's have a look at the two. If the black hair has the allele big B, and we say that that's for black hair, and brown hair is little b, then any two of the combinations, big B, little b, and big B, will code for this protein for black hair. Now, we say when you have both of the alleles that we have homozygous alleles. The way to remember that is homo just means the same. And if we have big B, little b, that is heterozygous. and hetero just means different. Now, we call these sets of alleles that you have the genotype. And then we call the trait that's displayed the phenotype. Now, one way that you can tell whether the allele is dominant or recessive is you can look for whether the letter is capitalized. A capital letter means that it's dominant, and a lowercase letter means that it's recessive. Now, something important to remember about recessive genes is you need 2 of them for that trait to be shown. So you need the one from your mother and your father in order for that trait to be shown. Now, we can use Punnett squares in order to work out the chance of someone displaying a certain trait. Now, they can use weird examples for these Punnett squares using genes that code for proteins you may have never heard of. So I'm just going to use a pretty obscure example that I use with my classes, and I'm going to use one of my favorite characters from Harry Potter to explain it. So if we take Hermione Granger, for example, and she's described in Harry Potter as being a mudblood, meaning that she has muggle parents, which are non-wizards. This means that her parents do not show the wizard trait, but they must carry it for Hermione to have been a wizard herself. So this means that they are both heterozygous carriers. And what we do with that is they must show then W, small w, must be the alleles that they carry from the mother and the father. And what we do is we put them into our Punnett square, like so, and then we can work out the chance that Hermione was a wizard. And the way I draw my Punnett squares is I go down, so I put in the letters going down first in columns before going across. So let's do that then. So big W will go in here, and big W will go in here, little w will go in here, and little w will go in here. And then I go across, so I've got big W here, big W here, little w here, and little w here. So if we look at this, Hermione only had a 25% chance that she would be a wizard, because this is the only pair of alleles that would code for the protein for her to be a wizard. If we look at the chances of her becoming non-wizards, well, we have 75% chance that she would be a non-wizard, and we have a 50% chance she would be a carrier of the wizard gene. So this is like her parents, she would be a heterozygous. We call them carriers where you can pass on the gene, but you don't show that trait yourself. And then she also has a chance of being completely a non-wizard. This would be a homozygous non-wizard. Physical characteristics are not the only things that are passed down through genetics. Sometimes genetic diseases are inherited as well. We're now going to look at a genetic disease called polydactyly. And polydactyly, it is a disease that can cause you to be born with extra fingers or toes. And polydactyly is a dominant allele. So if we have a look, if one parent shows polydactyly and they have the homozygous polydactyly, that would mean that they would have capital D, capital D. And if another parent does not have polydactyly at all, then that would mean they'd have to have both alleles that code for not polydactyly. Then if I complete the Punnett square here, big D, big D, big D, D, and then small, uncapitalized, going in there, then if you have a look at that Punnett square, then you will see that there is no chance that a child could be born without polydactyly if you have a homozygous polydactyly parent. We're now just going to talk about organization in a ecosystem. And to represent organization, here I have a food chain, and materials move through this food web when they are consumed. So the arrows are representing the energy of that food when it is consumed by something else. Now, it's important to remember that all food chains and food webs start off with producers. And if we look down here, we have two different types of producers. We have grass and a tree here. The reason why every food web or food chain starts with a producer is because they convert the sun's energy by photosynthesis into glucose, and without any producers, we would get no food webs or food chains. It also means that there is the most of this in our ecosystems, so there is more producers than anything else in our ecosystem, or they would not be able to support all the life. After our producers, we have our primary consumers, and our primary consumers eat the producer, and that means that energy gets transferred from the producer to that primary consumer. After primary consumers, we have our secondary consumers, and our secondary consumers are things that eat our primary consumers. And then finally, we have our tertiary consumers, which eat secondary consumers and some primary consumers, but they do not eat producers. Let's turn this food web into a food chain so we can look at some of the energy that gets transferred. So if I start with my producer here, which is grass, and I just go and follow one chain. If I go down this one here, I see my grass is eaten by, I think this is just a regular ant. And then after my ant, I have an aardvark. Then after my aardvark, I have a hyena. And then the hyena is eaten by a lion. Now, we can also call this lion here, it would be a tertiary consumer, but it'd also be our apex predator. This is a term that we use for organisms that aren't predated on themselves, so they are not prey species. All the other organisms that are eaten are called prey species. Now, the relationship between predator and prey in the ecosystems is quite complex, and here is a graph showing how the hare and Canadian lynx populations have varied between the years 1850 and 1930. And what you'll notice is that there is a bit of lag between the hare numbers increasing and the Canadian lynxes increasing after. You'll notice when the hare's population is massive here in 1865, it's taken a few years for the Canadian lynx population to increase as well. But basically the main things is that when the preys increased, the predators also increase because they have more food for them to eat. But there is always that 2-year lag where it takes the predators a few years to catch up. And if we look when the prey numbers decrease rapidly here, this could be caused maybe by a disease to the hare population. You can see it drops incredibly low here. It takes a few years for the lynx population to drop after that. And the prey's number could be controlled by a number of different things in their ecosystem, between biotic factors, for example, the introduction of a new predator species, to abiotic factors, things that they can't control, like the weather or the change in soil pH not producing enough producer. When organisms die in our ecosystems, the materials are recycled, and we call this recycling decomposition. And there are some key players in decomposition that recycle the nutrients. And the main decomposers are bacteria and fungi. An example of a key nutrient that is recycled in our ecosystem is carbon. So what happens with this carbon is the bacteria and fungi break down the longer chain molecules in the organism and break it down into smaller molecules which can be absorbed into the ground when there's enough waterfall. Remember, there is a finite amount of atoms in this universe. Nothing can be created or destroyed, so they are just recycled into the ground. We call how fast the decay happens the rate of decay. And the rate of decay is controlled by three main factors, the first being temperature. Now, it's important to remember that enzymes are involved in breaking down the organism, and obviously enzymes work at an optimum temperature. It's probably going to be around 25, 26 degrees, and this is when enzymes work their best at breaking things down. Obviously, if it was too hot, then the enzymes would denature, and if it was too cold, then the decay would happen much slower. The second factor that can affect the rate of decay is the availability of oxygen. The reason why the amount of oxygen can affect the rate of decay is because of the fact that the organism's bacteria and fungi need this oxygen in order to respire. And if there's more oxygen, they can respire faster and break down the organism faster. Finally, the rate of decay is also controlled by the amount of water available. Just think, decomposers require water in order to survive, in order to respire. So this is also going to be one of the factors that affects our rate of decay. So we looked at how carbon could be returned to the ground. It's not just carbon, there's nitrogen as well, which is another key mineral which plants need to survive. But let's look at the full cycle of what's going on here. Let's look at the carbon cycle, to how carbon is put into our atmosphere and is taken out of our atmosphere and back into the ground. So let's start off where we just left and looking at that decay and how these animals, when they die, or organisms like plants, return carbon to the ground. Well, how does that carbon leave the ground? Well, it can leave in a few ways. For example, if a plant takes up that carbon as it grows and then is eaten by an animal, well, that animal is going to respire, and respiration is a key way that carbon dioxide is put into our atmosphere. As well as this, we also burn fossil fuels. The burning of fossil fuels is something that is disrupting our carbon cycle at the minute because of the fact fossil fuels have taken millions and millions of years to form, and there is so much stored carbon in our ground, and we are returning it to the atmosphere now, and trees cannot remove it at a quick enough rate. So what ways are then carbon, once it's been put into the atmosphere, removed? One key way that carbon is removed from our atmosphere is through photosynthesis. Photo means with light, synthesis means to make, and what it does is turn this carbon into glucose, which the plant uses as food. And remember, if that plant is eaten by an animal, that carbon is then transferred to that animal. Or if that plant dies, then decomposers will break it down and store it in the ground. We also do have some locked carbon, which is stored in rocks, which you know from your GCSE chemistry as well. One other material that we need to know how it cycles around in our environment is water. So let's have a look at this water cycle, and we'll start off in the ocean. And evaporation happens from water in our ocean. This is where it gets turned from our liquid state into a gaseous state and travels up into our atmosphere. We also get some water evaporating from plants, and we call this transpiration. I'm going to remember from biology paper 1, we looked at the factors that can affect transpiration. How hot it is, the humidity, and the wind speed affects how quickly this transpiration happens. And then once our water has evaporated, it will rise into our atmosphere until it starts to cool. And when it is cooled significantly enough, we get precipitation. And precipitation is when water condenses and it rains down onto our ground. Now, some of that water is used up by trees and other plant species. However, a lot of it filters into the ground. We call this infiltration, and some of it gets stored in groundwater. But a lot of it travels through rivers all the way back to the ocean, where this cycle repeats itself again. Now, this is the end of today's video. Thank you for watching. Remember, if you did like it, please drop it a like and subscribe to the channel. And thanks again for watching this video.